So our next speaker is Ben Knollenberg. Ben is a currently, okay, let me just undo this for 1 second. I need to spotlight Ben.
Go down on the bottom. There.
Hold on.
To select him first.
Ben. Spotlight for everyone. There you go. So Ben is a currently scientist at the Wrigley Cocoa Genetics and Breeding Unit in Davis, California. And he's going to talk to us a little bit more about the his work during his postdoc at Penn State. As you see, we're trying to decipher more the the response to Phytophthora in cacao. As you all know, cacao, Phytophthora is one of the worst and the widespread pathogens of cacao, and so Ben's going to talk a little bit more about it. And this work is in collaboration with our partners in CIRAD and also in CEPLAC in Brazil. So go on, Ben.
Yeah. Yep. So as Ciela mentioned, I'm with Mars Wrigley now, but this is going to be mostly work from my PhD and a little bit from the postdoc that I did with Mark and Ciela at Penn State. And during my PhD, I stumbled upon this compound called Clovamide, which seemed to be important for differentiating tolerant and susceptible clones that I was looking at. And then after that, I started digging into the biosynthetic pathway using transcriptomics, more metabolomics, and a little bit of QTL analysis. And was able to elucidate a couple of the steps in the pathway in cacao for biosynthesis of clovamide. And I want to contrast that with the pathway that's been described for red clover. So both red clover and cacao both make the same compound, clovamide, but I'm hoping that I can convince you today that they make them in different ways, and it's an example of convergent evolution. So I'm going to briefly go over my intro slides, which I think everybody has covered pretty well so far, so I can skip through those. And then I'll talk about the transcriptomics and metabolomics work and some of the gene functional characterization stuff, and then a little bit on QTL analysis to identify regions of the cacao genome associated with clovamide abundance. So I think everybody knows the importance of cacao. It's, a huge industry, and it contributes to the livelihood of many farmers, but there's this huge limitation at the beginning of the supply chain with limited yields. And one major contributor to the limited yields is disease. And we've seen this table already. Black pod rot is the major disease of cacao and is caused by several species of Phytophthora. So there's a lot of work that the group at Penn State has been doing, and you've seen this already in Noah's presentation, so that's great. Don't have to get into it too much, but we have a broad range of tolerance and susceptibility. It's quantitative resistance with high and low tolerance and everything in between. And for my PhD work, I decided to focus on two clones, one that is very tolerant to black pod rot and several other cacao pathogens and diseases, and one that is very susceptible. So Scavina 6 has been used for decades in breeding programs for its sort of broad spectrum resistance to cacao pathogens, and ICS1 served as the susceptible control in the work that I did. And I just asked the very basic question, does Scavina 6 have something that ICS1 does not, at the level of metabolites, that is? And to address this, I did some metabolomics using liquid chromatography and tandem mass spectrometry, or LC-MS-MS. And since that disease resistance phenotyping data that you saw just before was done with the stage C leaves, this kind of intermediate developmental stage, I stuck with that stage for my first comparison. And I just compared basal metabolite profiles of these two clones, Scavina 6 and ICS1. And we detected a bunch of metabolites, 1,719. And you can see here, this is the mass to charge ratio and the retention time in the chromatogram from the HPLC with tandem mass spec. And any peak with a green dot is higher in Scavena 6. Any peak with the red dot is higher in ICS1. And I applied some filters to this data set so that we can kind of pull out the metabolites that seem to be most important for contributing to Scavena 6's resistance potentially. So the filters that I applied were that the metabolite had to be higher in Scavena 6 by at least 5-fold. And that, ended up giving us 183 metabolites. And then of those, I just took the 30 most abundant metabolites in Scavena 6. And I thought this was, a good way to pare down the data set and help us zero in on some metabolites that might be important. And here are those top 30 metabolites that we ended up paring the data set down to. And you can see there's like little tiny signal for ICS1 and a large signal for Scavena 6 for all of these. And these are all ions that were detected in mass spec. And one thing that happens with mass spec is that you can get multiple ions for the same compound. So every signal here with a blue arrow is associated with actually a single compound. And that is Clovamide, shown here, so this phenolic compound. And it was the most abundant signal in Scavena 6 based on the mass spec signal, and it accounted for about 7% of the metabolome signal. And it was about 40-fold higher in Scavena 6 than in ICS1 if you combine the intensity of all the peaks associated with that compound. So to give you a little bit more background on this compound, it's a hydroxycinnamic acid amide. So that means it has a hydroxycinnamic acid moiety, in this case, caffeic acid, and it has an amino acid moiety, in this case, L-dopa, and they are stuck together with an amide bond in the middle. And you can imagine that there are several variations on this theme. You can vary the hydroxycinnamic acid group, and you can vary the amino acid group and get all these different combinations. And here in the middle is caffeic acid and dopa, giving caffeoyl dopa, which is another name for clovamide with the red star. And if we go digging in the LCMS data set, we find that we can find a bunch of these HCAA compounds, and all of them that we detected in this table were higher in Scavena 6 than in ICS1 quite substantially. And we found it for several other HCAA compounds as well. So this table is a little bit expanded in the publication, but I'm just showing a limited subset here. So we decided to focus on Clovamide because, again, it was the most abundant signal in the Scavena 6 extracts, and it was about 40-fold higher in Scavena 6 than ICS1. And when we went to the literature, there's a lot of previous work that's been done on HCAAs, identifying them as important contributors to resistance in several different plant species against several different pathogens, bacteria, fungi, and oomycetes. So they come up a lot in the literature as important contributors, either as antimicrobial compounds directly or antioxidant compounds or cell wall reinforcers, because they can do phenolic oxidative coupling with lignin and stuff like that. So we did a little bit more targeted quantification of the clovamide. So you can see here an HPLC diode array detector chromatogram, and here's a big old peak for clovamide in the Scavena 6 extract, and you can not really see it unless you zoom in super far on the ICS1 extract. And here's the standard for clovamide. And since we did it this way, we can make a standard curve and get absolute quantification, like milligrams clovamide per gram of tissue, rather than kind of like a arbitrary mass spec count relative abundance. So we see that in the young, the middle age, and the old leaves, there's a lot more clovamide in Scavena 6 than in ICS1. So it's throughout leaf development. And, we want to determine if this is actually important or if it's just a molecule hanging out doing nothing. And so we had some clovamide synthesized with some collaborators in the chemistry department at Penn State and put the compound in some media and grew 3 different pathogens on it: Phytophthora megakarya, palmivora, and Tropicalis. And you saw this in Noah's presentation as well. And it inhibited growth of all 3 of those Phytophthora species that contribute to black pod rot in cacao. And in this assay, I used 2 millimolar clovamide, and that's well within the physiologically relevant range that we see in the cocoa tissues. So in some of the Scavena 6 leaf extracts, or in the tissue, we saw up to 11 millimolar clovamide. So, we are limited by certain experimental factors in this assay, but we settled on 2 millimolar because it was physiologically relevant. Also in this work, I found that clovamide inhibits proteinase K. It also inhibits pectinase, and we know that proteases and pectinases are very important contributors to virulence for Phytophthora during infection of cacao tissues. And you can also see that in some of the work from the Bailey group, the transcriptomic analysis showing that there's a lot of proteases and pectinases that are expressed by Phytophthora when they infect cocoa tissues. And in this work, we also showed that clovamide is a polyphenol oxidase substrate. So that means it contributes to the browning reaction that is pretty ubiquitous across a lot of different plant species. So, you know, you've seen it in apple when you bite it and leave it out, and you can see it, you know, also in cacao being infected by Phytophthora. You have a little bit darker and smaller lesion in Scavena 6 and slightly less dark here in this picture. And that browning is from oxidation of the clovamide, and that contributes to the enzyme inhibition because the oxidized form can cross-link proteins. So yeah, we're interested in it because it's important for disease resistance in cacao, but it's also an interesting compound for human health because there's a lot of reports in the literature of clovamide being antimicrobial against human pathogens. It's anti-inflammatory and potentially neuroprotective. So there's, you know, a little bit of buzz around it in terms of human health stuff too. And here it is inhibiting plaque formation of beta amyloid protein, which is associated with Alzheimer's disease. This is an in vitro assay, but still pretty cool, pretty interesting. So, you know, it seems like an interesting compound, good for cacao disease resistance, potentially good for us. So we want to understand a little bit better what causes Scavena 6 to accumulate so much of it, whereas ICS1 doesn't accumulate very much of it at all in the leaf tissue. And to address this, we did some transcriptomics and metabolomics to identify clovamide biosynthetic gene candidates. So here's an overview of the experiment. I took young leaves, middle-aged leaves, and old leaves from the Scavena 6 and ICS1 genotypes. And for each of these sample groups, I got 5 biological replicates, so 30 samples total. We did RNA-seq and got relative expression data for over 20,000 genes. And then we did LCMS metabolomics and got relative abundance of over 1,000 metabolites. And, you know, before I get into any of that data, I just want to point out that's a lot of data. Like, where do you begin when you have expression for over 20,000 genes? I wanted to decide like which genes we should really focus on. And in order to do that, you have to think about how clovamide might be made. And the most probable pathway is an acylation reaction that takes caffeic acid and L-DOPA and sticks them together with an amide bond. And in order to make this happen, you'll need an activating enzyme to add an activating group to the caffeic acid. So that serves as a good leaving group. If you think back to organic chemistry class for like, SN2 reactions, you need a good leaving group. And then the L-DOPA can then do a nucleophilic attack on this activated form to make clovamide. And that last step is catalyzed by an acylating enzyme. And you have two main options for this type of acylation reaction in plant metabolism. Option 1 uses a UGT or a UDP glucose dependent glycosyltransferase for the activation step, and it adds a glucose as the leaving group. And then the acylation step is catalyzed by an SCPL-ATs. There's another option where the activating enzyme is 4CL, and the activating group that it adds is coenzyme A, and then the acylating enzyme is a BAHD-acyltransferase or BAHD-AT. So this is a common dilemma that comes up when people are trying to figure out how acylations are performed in plant metabolism. And here's a review you can check out. What a dilemma, if you want to learn more about the topic. And we already know that in red clover, this BAHD-acyltransferase pathway is the route that it takes to make clovamide. So it uses 4CL, adds a coenzyme A activating group, and then uses the BAHD-acyltransferase to do the acylation. But we don't really know in cacao. So in order to get some clues, I used the metabolomics data to look for trade-offs in the metabolome. So remember, we got relative abundance for over 1,000 metabolites. So I took the abundance of clovamide and correlated it with every metabolite in the data set, so 1,313. And then I sorted by the most negative Pearson correlation coefficient and generated this table. So these are the compounds with the strongest negative correlation with clovamide. You can see the Pearson correlation coefficient and the p-value here. And just briefly, we have luteolin glucoside, quercetin glucose acetate. We have apigenin glucoside, and we have campherol glucoside. So we have a lot of flavonoid glucosides. So it looks like there's a negative correlation between clovamide and flavonoid glucosides. And I thought that this might provide a clue as to which pathway cacao uses to generate clovamide. And so this over here on the right side is that UGT SCPL option that I showed before. And we know that UGT enzymes use a co-substrate called UDP glucose. So if scavina-6, hypothetically, is using a UGT pathway, it's going to be hogging a bunch of UDP glucose relative to ICS1 to make clovamide. So there's going to be less UDP glucose available for flavonoid glycosylation. So luteolin to luteolin glucoside, for example, because you add a glucose here. And both UGTs for both of these reactions are going to need this co-substrate. So maybe that's why we see this much higher accumulation of flavonoid glucosides in ICS1 relative to scavina-6, whereas scavina-6 makes a bunch of clovamide. And again, this is in contrast to the BAHD-acyltransferase pathway, where there is no glucose involvement at all. So based on that little nugget, I decided to focus on UGTs and SCPLs in the gene expression data. And in the young leaves, we had over 1,000 differentially expressed genes, same in the middle-aged leaves. And in the older leaves, there were over 6,000 differentially expressed genes between the two clones. But now that we have some targeted gene families, we can kind of zero in on certain candidates. And I identified candidates for each of the steps that we're interested in by taking known protein sequences for proteins that catalyze reactions or similar reactions, and then blasting them against the cacao genome. And for the UGT step, I got 134 candidates. And for the SCPL step, I got 34 candidates just based on this amino acid blast alone. And so we have expression data for young, middle, and old leaves. And I'm only showing the 50 UGTs here with the highest expression. And I have them sorted by their blast score, their E value. And I ended up picking these three down here because they all had higher expression in scavina-6 than ICS1. But if you look at this rank, this is their rank in the blast for like best blast hit using the known function UGT. And based on sequence alone, there were over 100 candidates that were better based on the blast score, but I chose these based on differential expression. So here they are, kind of zoomed in on each candidate gene for this UGT step. They're all expressed a bit more highly in scavina-6 than ICS1, at least in the young leaves for this one, and then they kind of even out in the older leaves. And I did the same thing for SCPL, but with the SCPL step of the pathway, there was really nothing interesting in terms of differential expression. So I just went with raw expression, like which were the most highly expressed. And I chose 3 candidates just based on being the most highly expressed SCPLs. So I have 3 UGT candidates and I have 3 SCPL candidates. One of them had 2 pretty distinct alleles, so I kept track of both of them. I cloned the cDNAs and I put them in overexpression vectors. And I did kind of a combinatorial approach, doing co-expression of a UGT with an SCPL, and did combinations A through L. And I expressed them in a Cotiana benthamiana with this GFP marker gene. And then I punched out leaf discs where the marker gene was expressing, extracted metabolites, and looked for a clovamide formation. And luckily, one of the combinations, combination E, formed clovamide. So that was this UGT and this SCPL, that one. And then here I just have the control, which is the BAHD-acyltransferase from Clover. So thanks to Michael Sullivan from the USDA research station in Madison, Wisconsin for sharing that coding sequence. But so yeah, we found a UGT and an SCPL that do the job. So that represents an example of convergent evolution. We have the BAHD-acyltransferase pathway in red clover, and we have the UGT-SCPL pathway in cacao. And there is no orthology between the SCPLs and the BAHD-acyltransferases. And just as some supporting information, this UGT here, I sequenced cDNAs from Scavina6 and ICS1. And ICS1 has a 1 base pair deletion pretty early on that causes a frame shift, which leads to a pretty early premature stop codon. And that truncates the protein so that there is now this UDP glucose dependent glycosyltransferase, this UGT conserved domain that is kind of cut off from the protein, at least for the ICS1 allele. So here's the pathway again. We have higher expression of this UGT step in Scavina6, and we have a truncated allele in ICS1. And just briefly, for a bit more support, I'm going to show some QTL analysis results. And this was to identify regions of the cacao genome associated with clovamide abundance in young leaves. And luckily, an F2 population already existed at, in CIRAD, or sorry, CEPLAC in Brazil. And collaborators at CIRAD had already generated a nice map for this population. So I just had to go to Brazil and collect some leaf discs and then bring it back to Penn State and quantify clovamide in them. And I did that. And here you can see the clovamide abundance in the F2 population with the high accumulator parent, Scavina6, the low accumulator parent, ICS1, and here's the F1 parent, TSH516. And then when you do the QTL analysis, there is a very clear, strong signal on chromosome 4. And the signal is only 2 SNPs, and one of the SNPs was within 1,000 bases of the start codon of the UGT that we identified in the earlier steps. So it was a good bit of support for the UGT that we identified from transcriptomics and the expression in Nicotiana benthamiana experiment. So just to summarize the evidence for the UGT SCPL pathway being used by cacao to make clovamide, we had a negative correlation between clovamide and flavonoid glucosides. And that kind of implied UDP glucose competition between the two pathways, which suggested a UGT involvement. We had some UGTs with higher expression in Scavina6 and ICS1. And then co-expression of a UGT and an SCPL in Nicotiana benthamiana resulted in clovamide formation. And then we had a premature stop codon in the UGT allele for one of the steps here. In ICS1, it had a truncated version of the protein, predicted protein. And then also there's a QTL on chromosome 4, which supported the UGT candidate that we identified using transcriptomics. So there it is. This is the pathway as we know it right now, but there's some details that we still need to work out. But yeah, so there's an example of convergent evolution in a polyphenolic compounds biosynthesis between cacao and clover. And I'd like to thank everybody up here on the screen that helped out with the project. And also the funding sources and everybody else in the Gildan Maximova lab that helped with this project and with a lot of fun discussions along the way.
Thank you, Ben.
Awesome.
We do have a couple of minutes. Are there any questions? Please unmute yourself and just speak up.
Yeah, and so hi, Ben. It's a nice work. I'm Shaheen from Wellsville. So I have a question, like, have you also checked other tolerant varieties of cacao, like Pound 7, whether they have higher level of clovamide? And that's one question. And my second question is, like, you have checked the leaf, but I don't know. I think I have seen your paper way back, and you mentioned something about whether it's also high in the pod. Is there any difference between pod and leaf in terms of clovamide? So,
yeah, hi, Shaheen. Thanks. Yeah, so I looked at leaf tissue from the genotypes that were mentioned in Noah's presentation, like the NSF panel. and even among the resistant clones in that group, it seems like the high clovamide content in the leaf tissue is really specifically a Scavina 6 thing. So yeah, pound 7 was not one of the, did not have high clovamide, at least at basal level. I didn't quantify the induced levels of clovamide, but for basal clovamide level, Scavina 6 is quite unique. And then for pods, the two genotypes that I mentioned today in the talk, they both do have a lot of clovamide in the pod exocarp. So I measured it there, and I also did metabolomics on those tissues. And yeah, so they both have a lot of clovamide in the pod exocarp, but the difference in that tissue is that Scavina 6 has a bunch of sulfated clovamide and sulfated and methylated clovamide. So yeah, that seems to be the differentiating compound in the pod tissue.
Thanks again, Ben. We need to keep moving so we can keep our schedule.